Today we are going to discuss about differences between biological catalyst and chemical catalyst. So biological catalysis. First of all, biological catalysis means enzymes are the biological catalyst. Enzymes which can increases the rate of reaction without involving the reaction. These are the biological catalyst. So what are the differences means in chemical catalysts are the substances that increase or decrease the rate of chemical reaction but remain unchanged. But in case of biological catalysis, enzymes are protein in nature that increases the rate of chemical reaction, and these are used in the converting of substrate into product. So this is the first difference in between biological and chemical catalysis. And the second one, molecular weight. So chemical catalysts are low molecular weight compounds when compared to enzymes or biological catalysts are high molecular weight globular proteins. So next one, chemical catalysts are mainly two types. First one positive and the next one negative catalysts. So in the biological catalysts there are two types, activation enzymes and inhibiting enzymes. Next, nature. Nature of chemical catalysts are simple inorganic molecules. So the chemical catalysts which are used in chemical reactions are simple and inorganic molecules, whereas enzymes are complex molecules. That is nothing but enzymes are protein in nature. So they contain primary, secondary, tertiary and quaternary different structural organization levels. So that's why enzymes are complex molecules comparative to chemical catalysts. Next one, chemical catalysts are inorganic catalysts, whereas enzymes are organic catalyst or biocatalyst. Then in chemical catalysis, typically slower reaction rates are occurred in typically slower, but in enzymes more than faster comparative to chemical catalysis. For example, protein is taken in vitro conditions or laboratory conditions. Protein breakdown is carried out in the presence of chemical catalyst for several hours. But in case of in vivo conditions, in the presence of biological catalyst, it will take nearly 3 to 4 hours. So, based on this, reaction rates are typically slower in chemical catalyst, but in case of enzymatic catalysis, reaction rates are completely faster. Next one, chemical catalysts are not specific and therefore end up producing reduced residues with errors. But enzymes are highly specific in nature, so they can react with only specific substrates and produce relative products. Next, chemical catalyst or chemical catalysis process occurs in high temperatures and pressures, but enzymatic catalysis occurs in mild conditions, physiological pH and temperatures. So in chemical catalysis, carbon-carbon and carbon-hydrogen bonds are absent. But enzymatic catalysis, these carbon-carbon and carbon-hydrogen bonds are present. Why? Because enzymes are mainly protein in nature. So that's why proteins contain carbon-carbon bonds and carbon-hydrogen bonds. Then examples for chemical catalysts are vanadium oxide, and enzymatic catalysts are amylase and lipases. Next one, activation energy. So in the chemical catalysis, activation energy is lowest, and in the enzymatic catalysis, activation energy is also lowest. But in comparative to chemical catalysis, enzymatic catalysis, activation energy is lower. So these are the differences of biological and chemical catalysis. Enzymes. Enzymes are the biocatalysts, means the catalyst of life. A catalyst is defined as a substance that increases the velocity or rate of chemical reaction, but without itself undergoing any change in the overall process. Next, enzymes may be defined as biocatalyst synthesized by living cells. They are protein in nature except RNases. RNases which are RNA in nature. Ribozymes. These are called as ribozymes. And enzymes are colloidal, thermolabile in character and specific in their action. Example, in the laboratory, hydrolysis of proteins by a strong acid at 100 degrees centigrade takes at least a couple of days. But in the presence of enzymes, these are digested by using 37 degrees centigrade with a couple of hours. So by this example, we can understand the enzymes are biocatalyst. It can increases the rate of reaction and less amount of activation energy and decreasing the time of digestion. Coming to historical background, Berzelius, scientist in 1836, coined the term catalysis. 1878, Kuhne used the word enzyme. Next, the term substrate was introduced by Duklas in 1883. Yeast enzyme. In yeast, Buchner, James enzyme was extracted. The next, James Sumner isolate the enzyme urease. And let's coming to nomenclature and classification of enzymes. In the early days, the enzymes were given names by the discoverers in an arbitrary manner. For example, pepsin and trypsin and chymotrypsin convey no information about the function of the enzyme or the nature of the substrate on which they act. Next, sometimes some enzymes are contain suffix ase. Example, lipases and nucleases. Next, lipases which are act on lipids, nucleases which are act on nucleic acids. One more example is lactase on lactose. After that, trivial names are given to the particular enzymes, which however fail to give complete information of enzyme reaction. Next one, enzymes are sometimes considered under two broad categories. First one, intracellular enzymes and extracellular enzymes. Intracellular enzymes are functional within the cells where they are synthesized. But extracellular enzymes are active outside the cell. Examples, digestive enzymes. Next, International Union of Biochemistry appointed an enzyme commission in 1961. In 1964, the IUB system of enzyme classification has been enforced. Based on this, enzymes are classified into 6 major classes. So first one, oxidoreductases. Next one, transferases. Third one, hydrolases. Fourth one, liases. Fifth one, isomerases. And sixth one, ligases. Then first one, oxidoreductases. These enzymes are involved in oxidation and reduction reactions also. That type of enzymes are called as oxidoreductases. Examples, alcohol dehydrogenase, cytochrome oxidase, and L and D amino acid oxidases. And next one, transferases. These enzymes are involved in the transfer of specific functional groups from one compound to another compound. Best example is the transaminases. Transaminases are the best example for the transferases enzymes. Next one, hydrolases. These enzymes are involved in the hydrolysis reactions like adding of water molecule. Examples, lipases, cholinesterases, acid and alkaline phosphatases, etc. And fourth one, liases. These enzymes are involved in the addition or removal of water, CO2 and ammonia, etc. Examples, aldolase, fumarase and histidase. Then aldolase enzyme which is involved in the glycolysis reactions. Next, fifth one, isomerases. These are the enzymes which are involved in the isomerization reactions, interconversion of two different isomers. Example, triose phosphate isomerase. This is the enzyme which is responsible for the interconversion of glycerolide 3-phosphate to dihydroxyacetone phosphate. This reaction is occurs in the glycolysis. And next example is the phosphohexose isomerase. This enzyme is also involved in the interconversion of glucose 6-phosphate to fructose 6-phosphate. And the final one is the ligases. So ligases are the enzymes which are involved in the combination of two different compounds with the utilization of ATP. Best example is the glutamine synthetase, acetyl-CoA carboxylase and succinate thiokinase. Here ATP is utilized to form ADP and phosphate.